Hello and welcome to BIOS Builders. We're absolutely thrilled to welcome Marion Dorsch and Jared Rutter to the show today. Thank you once again both for joining us.
We're very excited to be here today, and thanks for giving us this opportunity to tell you the Atavistic story.
Before we kick things off, however, Marion, can you share a brief personal introduction with us?
Yeah, happy to. I'm Marion Dorsch. I'm the President and Chief Scientific Officer of Atavistic Bio, and I was one of the founding members together with Jared here and with our second founder, Ralph de Berardinis. I'm a biologist by training. I got my PhD in Germany and then came to the US, first to New York City and then to Boston ever since. I have worked in biotech pharma for more than 20 years with a focus on small molecule drug discovery in oncology as well as in rare genetic diseases. And I have worked on a number of innovative therapies that are now approved by the FDA and are really making a difference for patients. So I'm hoping to do that all over again with this amazing team here at Atavistic.
I absolutely love that message and would love to dive in deeper in just a moment. But before we do, Jared, can you share a brief personal introduction as well?
Yeah, thanks, Chris. So I also am a biologist by training. I did my PhD at UT Southwestern with Steve McKnight, and that was really a formative experience for me. Steve was really a powerful mentor that empowered people in his lab to take on bold topics and ask bold questions. And I think that's really, I've tried to model that throughout the remainder of my career. I did a short postdoc there at UT Southwestern and then joined the faculty at the University of Utah in 2003 and have been here ever since and have built a lab really focused around the same kinds of questions we were asking in Steve's lab, 20 plus years ago, trying to understand metabolic sensing and the biochemical mechanisms whereby metabolites signal to change how cells behave. And I've had the good fortune of being able to be involved in a couple of ventures in the private sector, having founded a couple of companies and seen a couple of companies that I found to take small molecule drugs into the clinic. And that's been a really exciting experience. And hearing Marion introduce herself reminds me of how excited we were when she agreed to be the CSO of Atavistic, because she's obviously the right person to take this program forward.
And that's a perfect segue, Jared. We'd love to understand, even just briefly, more on the underlying technology that led to Atavistic's genesis, or maybe said in another way, what attracted you to spin out and build Atavistic in the beginning?
Yeah, I mean, so as I alluded to, one of the questions that my lab has had, that I've had as a scientist for more than 20 years, is how do metabolites signal to change how cells behave? There's a lot of evidence that this happens, but we really just don't have great technologies for discovering the interactions that drive this signaling or that underlie this signaling. And so over the last several years in particular, we've been focused on developing technologies to enable the discovery of those interactions. And one of those technologies that was developed by Kevin Hicks, who at the time was a postdoctoral fellow in my lab, is a technology that we called MIDAS, which is a very simple technology based on simple and robust biophysical principles related to equilibrium dialysis that enables us to discover metabolite protein interactions with very high sensitivity and a lot of robustness. And that technology, we developed it, as you might imagine, just trying to answer fundamental biological questions. And it became clear pretty early that if this worked really well, this technology might have the ability to identify small molecule binding sites on proteins that might be of therapeutic interest. And if we could discover those binding sites, that might be establish a beachhead that could be used to develop therapeutics that act on those sites. So that's sort of where it came from. Again, trying to understand basic, cellular biochemistry, try to fill a technological gap that we were concerned by, and that kind of led to the technology that founded Atavistic.
Maybe at some point during our conversation, we can send you to the whiteboard behind us to guide us through single initial technological detail. But for right now, Marion, with that context, could you dive a little bit deeper and just provide an introduction to Atavistic for us before maybe telling us a little bit more about its current status?
Yeah, so Atavistic Bio is a startup in Cambridge, Massachusetts, and we were founded in 2021 in the mid of the pandemic. We are trying to leverage, or we are leveraging this great platform that Jared has developed to map protein metabolite interactions to really discover novel allosteric small molecule therapies that can be transformative in a variety of diseases. And to do this, we really have adopted the technology here at Atavistic. We have optimized it further, and we're using it in a way, and we have renamed it to AMS, which stands for Atavistic Metabolite Protein Screening. So MIDAS is now AMS. So you will hear a lot of AMS. And we're leveraging that so that metabolite can be the bait to find those novel regulatory sites on proteins. And then once we have identified those regulatory sites, we use that as a starting point for small molecule drug discovery to discover potent and selective small molecule inhibitors against it. And this is really a very unique way to go after targets that historically have been difficult to target. They're called undruggables or hard to drug. And we feel like this new approach we are using here for the first time really opens up a scalable and systematic way to find ways to tackle difficult targets. Our initial focus is in oncology and rare diseases.
And as we think about, please, apologies, continue.
Oh, no, I just wanted to say, because of this focus also on rare diseases, that's where our second founder, Ralph de Berardinis, comes into play.
Oh, if I know he's not here to join us today, but in that case, I know you've mentioned Ralph twice. Is there any way you would be open to sharing a little bit about his background and rounding off that founding team?
Yeah, he is a physician scientist, a professor at UT Southwestern, and just really an expert in those rare genetic diseases in one areas of metabolism. Really a walking encyclopedia, and he has been incredibly helpful helping us to think through what diseases to go after and what targets to go after. So it's this group of folks that founded a company that's amazing.
It sounds like an incredible and complementary team. And as we think about that and maybe take a step back, we've talked about the technology, we've talked about a little bit, at least about your past. Would love to learn more about the actual genesis of Atavistic. What was the spark that started it all, Marion? And given your success in your career thus far, what really motivated you to join and lead Atavistic as CSO?
Yeah, so for me, I mean, when I look for a new opportunity, there are three things that are incredibly important. I mean, one is the underlying science. It needs to make sense. And I need to be convinced that it can really lead to novel therapies and innovative findings. And so clearly, the platform here, the technology checked that box, and our founders were really a great incentive to join the company. The second thing is the team. In this case, I had the opportunity to build the team from scratch. So I was able to really hire the best and most talented people to get the company going. And the third aspect is obviously the financial backing. And here, I'm very happy to say that the Column Group, which initially worked with me and Jared and Ralph to get this company started, is really known for their investments in great science and spinning out great companies. And they were joined by two other great venture firms, such as Lux Capital and Nextech. So all those three factors really came together, and it was just at the right time for me to jump in to get started.
Oh, it sounds like the perfect fit. And Jared, from your perspective, was there any specific spark? It sounds like with what was MITRE technology being ready to go and bringing on those right folks, it seemed like a good fit. But was there any background, any fun story to include from your side as well?
Yeah, there actually is. So, I'm not averse to starting companies, as I kind of alluded to, and I would like to think I'm creative about this and insightful, but I hadn't thought of it in the context of this MIDAS technology that we were developing. It felt like, there were a couple of reasons that I just didn't see it. And I was giving I had given a talk at a conference, and one of the advisors to the column group, Helen Hobbs, had seen this talk and had told the column group, "Hey, I think this technology might have legs in a company." And so I got a call from Tim Kutsky, who's managing partner at the column group, and said, "Hey, I want to talk to you about this." And I said, "Tim, look, I love your firm. I think it would be awesome to start a company, but I just don't know if this is the right technology for it." And he basically talked me into it, and man, I'm so glad he did, because it's been such a fun experience and such a success. But I think we were talking a little bit offline about the how things from academia are sometimes lost in translation as they enter the private sector, and this is a case where I thought I understood that translatability paradigm, but Tim definitely educated me and recruited a great CSO in Marion, and it's fun to see where Atavistic's going.
Oh, and there's always more to learn, and it also seems like now you're ready for that next time. So that way you've learned the lesson, and we'll see what's coming next. But as we talk more about Atavistic today, Marion, you're a seasoned executive with more than 20 years of biotech and pharma experience in oncology, inflammation, rare genetic diseases, formerly CSO at Blueprint Medicines. As we think about the context of that experience, can you share more about some of the issues and challenges we face broadly in drug development? Maybe what are some of those key unmet needs, and how Atavistic is starting to address some of these challenges?
Yeah, so actually for the longest time, many biotechs and pharma companies were really very focused on finding the next great disease target to go after. It turns out there are many great targets already known, but they are difficult to drug. So they don't have necessarily a clear pocket that one can go after with a small molecule, or they are just they don't have an active site, they're not enzymes. And so I think what is really needed are novel ways to go after those undruggable or historically difficult to drug targets. And I think allostery is clearly a way to go about that. And I feel with our platform, where we can use metabolites as baits to find novel allosteric sites on proteins that then we can leverage for small molecule drug discovery, that really opens a door into going after those difficult targets that are clearly genetically validated, and sometimes even clinically validated, but then there are issues that have arisen because of resistance development. And so one example, P53, a very the holy grail in oncology, a target that everybody would like to go after. So I think with this protein metabolite interaction platform that we're using, we really have a unique opportunity to try to tackle those more difficult targets. And that's what we really have set up. I mean, without going into any specific targets we're working on, I mean, the majority we are pursuing are either genetically validated or clinically validated. And there's just a unique angle to them. And so far, this really has worked really well. I mean, not only were we able to establish the platform at Atavistic, we also have improved it and made it more robust, because we're doing it at a higher scale than Jared. I mean, Jared had done an outstanding job doing it. And we also, in addition, were able, thanks to the great team of experienced drug developers that we hired here, to integrate it with our structure-based design capabilities, data analytics, informatics, and computational modeling, and really leverage it more towards a drug discovery angle that helps us find novel allosteric modulators for those difficult targets.
And just as a quick step back for those who might in our audience be unfamiliar, allostery tends to refer to the processes where binding at one site of a biological molecule affects binding activity at another site. So you're able to control and regulate that corresponding function a bit more. And that exactly. Part leads us to exactly what you were describing about those metabolite protein interactions, I think, which is also where I actually wanted to ask, how in your mind and why is this technology so promising compared to alternatives in the current drug development stack, especially for things like oncology and inborn errors of metabolism or rare genetic disorders?
I think what we're trying to do really at Atavistic Bio, and what Jared started doing in his lab, we are trying really to learn from biological networks that have evolved over millions of years, where proteins in a cellular context are regulated by the environment. And often that environment in a cell are those endogenous metabolites that are kind of breakdown products of macromolecules. And so there are many examples already of that in the canonical metabolic pathways where metabolites can regulate proteins. So now we have this opportunity for the first time to systematically really leverage that and map those protein metabolite interactions. That wasn't possible before, because this is a very scalable and systematic approach now to do. So it's a more empirical approach. If you think of other companies that are looking for allosteric sites, often that's more computationally based or using different approaches. So in our case, it's really empirical. And very frequently what we have found, and Jared has seen that also in his studies, is that the metabolites have a functional impact on the protein. So not only can you use the metabolite to find your binding site on the protein, you can then immediately ask, does this binding event have an effect on the protein? Can it inhibit or activate it, depending on the therapeutic context? And this might be due to the physiological relevance of metabolites, that this is more often the case than not. And then from our perspective, once you know that you have a functional relevant binding event, this is worthwhile pursuing, and we trigger structural biology and other means to identify the nature of the site, which we then use for small molecule discovery with structure-based design, computational modeling, and other approaches. So I think it's really this combination of leveraging existing information in a cell, but kind of getting it out, extracting it in a way to use it and to translate it into actions. No, continue.
I was going to say, and then all of this comes together to form the Atavistic discovery platform, the AMPS platform that you mentioned earlier, with those computational approaches, assessing the empirical information to evaluate those metabolite protein interactions in a high throughput manner. Are you able to share a little bit more about that overarching platform? Are you doing these metabolite protein assessments in solution and then testing them within cells, scaling from there? How are you broadly assessing these interactions in the first place?
Yeah, I mean, Jared, do you want to give a little bit spiel on the underlying principle, how you find the interaction, and then I take it from there, how we follow up?
Yeah, that'd be great. So basically, it's a very simple principle, Chris, that the platform is basically designed, again, built around the principle of equilibrium dialysis, where a library of small molecule metabolites is separated across a dialysis membrane from a purified protein of interest, the target we're studying. Those metabolites can come to equilibrium across that membrane such that the free concentration of the metabolites becomes the same on both sides of the membrane, but any metabolite that binds directly to the protein of interest is then enriched in that chamber, the protein containing chamber, relative to the opposite chamber. And then it's just a matter of quantifying all the metabolites, and you're left with situations where, you know, a protein binds a metabolite, that metabolite is enriched, and you can see that empirically because of the change in concentration in metabolite. So that's basically the Midas AMPS platform in a nutshell. And then, you know, I think we've found, and Marion has found as well, and Atavistic has found as well, that the platform works to identify binding, as she will allude to now and has a little bit in the past few minutes. That's just the first step in then trying to take that knowledge and turn it into a small molecule that might be effective as a drug.
Yeah, no, as Jared described it, that's really just finding those metabolite bindings using the platform is the first step, and it's obviously the most important step. But then you need to be equipped to carefully follow up. And so we have built here this really great team of experienced drug hunters from different disciplines that helps us follow up. So as I mentioned earlier, once we have those metabolite binders, we set up biochemical and biophysical assays to evaluate if that binding event has an impact on the protein. And just to make it a little bit more real, let's say you have a diseased protein that has a mutation that makes it less active. And because of this decrease of activity, it leads to the disease. So here your goal is to restore the function of the protein. And so we then set up assays to measure if those metabolite binding events can lead to increased activity of the protein, restore it to its normal level. And then if we see that, we know, okay, this is a site we really want to, this is a binding event we want to pursue further. And that's when we trigger structural biology technologies, usually X-ray crystallography, but we can also tap into other technologies there to really ask where exactly on the protein does the metabolite bind? And how does this site look? Is that an allosteric site? Is it amenable to small molecule drug discovery? And if that checks all the boxes, that's when we really kick off our structure-based design capabilities here, our medicinal chemistry optimization to find small molecules that fit into that site, into that pocket, and can leverage it. And so the metabolite is not the drug. The metabolite is your tool to find the binding site, and then you use that knowledge to develop a small molecule therapeutic against that site. And then we also evaluate in many cases, we were able to show that the metabolite has cell-based activity. So we can show that it restores the function of the protein in a cell, for example, which is always very motivating and encouraging. So, I mean, we have screened so far a large number of proteins through AMPS, and it's really working. We were able to discover known sites on proteins, known regulatory sites, which is a very good thing. It validates the platform, and many novel ones that we can now pursue. But what is even, I mean, ultimately as a drug discovery company, our goal is to get new therapies to patients. So I'm very happy to share that we were able to advance 2 of our targets into the so-called drug discovery stage. So they're now in the process of identifying a drug candidate that then can be advanced to patients down the line. So they're in the active drug discovery phase. And we are only 2 years in existence. So I'm very pleased how far we have come. And I think that is really a great example how you use a platform that helps you to find those interactions, and then you translate that into something that can lead to novel therapies.
Oh, that's truly an incredible accomplishment. And as you mentioned, the platform, so if I was to quickly summarize what you both said, please correct me if I err. The platform works by evaluating these metabolite protein interactions, screening proteins against your proprietary metabolite library, and doing so to determine binding sites for biological relevance. This in turn forms an interactome map of protein metabolite databases, and then it can reveal the sort of crosstalk between those two that can be used to inform small molecule drug discovery and development.
That is the second aspect of
Second.
So building this protein interactome map is something we do as we screen a large number of proteins. And the goal there is that over time that will lead to novel biological discoveries of interfaces one can interfere with to alter the disease process. In the direct way that we're using right now at Atavistic is we're using really the metabolite as a bait or a homing beacon to identify the novel pocket on the protein. And then once we know how the metabolite binds into that pocket, that knowledge is really valuable for us then to do structure-based design of the novel chemical matter that fits into that pocket. Or in instances, we're doing computational modeling of small molecules that could fit into the pocket. And then we further optimize them to make them more potent and selective. So there are two aspects. So there is the drug discovery path, and then there is the finding new biology path, which is equally exciting. But that's something we're doing over time. Jared, you want to comment from your? I mean, I think Jared is leveraging it more for the latter. We're leveraging it more for the first.
That makes sense. And yeah, as you're doing so, that landscape, then that interactome map would be constantly growing more robust and more predictive with each set of data incorporated. And so, Jared, would love to hear your thoughts on the potential of the technology to reach a number of different therapeutic areas.
Yeah, Marion's done a great job of explaining the technology. Let me just make one additional point that she briefly alluded to, that the goal is not just to find sites on proteins where a small molecule can stick adventitiously. The idea is to find sites on proteins that have been evolved over millions of years to integrate, binding with functional changes in the protein that confer evolutionary advantage on the organism. This is real biology here that's happening in the cell all the time. We're not trying to do something that's never been done before. We're trying to do something that the cell normally does, but it just doesn't do it well enough in that disease context, and then leverage that information to make a drug that is safe and effective. And so I think that's, in my view, that's kind of a different way of thinking about drug discovery, where that's actually the origin of the name. This was another product of Tim Kutsky. Atavistic sort of relates to shadows of the ancient past that are visible today, that affect how things work today. And that's the idea, that we're trying to take, evolutionary time, the learnings of evolutionary time, and employ them in drug discovery today. And that's kind of the I'm really excited about the opportunity to do that. And to specifically get to your point about this interactome map, I mean, this is what we dream about in my lab. We talk about this all the time, of a big matrix where we can ask systematically the question for all proteins in the human proteome and the entire metabolome: what are the interactions between those two sets of molecules, and what are the effects of those interactions? And that is not a data set that we will be generating today or tomorrow. This will take a long time, but you can imagine, in the fullness of time, if we really knew that, and we knew it well, that would transform our ability to not only do drug discovery in the way that Marion's been describing, which I think would be powerful, but it would also teach us a lot just about the underlying logic of how cells work, and how communication between processes works inside cells, and how it goes awry in the context of different disease states. So yeah, there is a blue sky version of our thinking that extends to include that entire big matrix, that hopefully we will get to someday. But I think in the near term, Marion and her team are doing a great job of trying to take the interactions that they have and turn them into drugs.
Yeah, and as you can imagine, I mean, building that database of metabolite protein interactions, I mean, that's a lot of information that we're collecting. So we really had to invest quite a bit in having a very good data analysis pipeline, and also ways to mine those data. And also over time, as we add data over the years, that we are able to look at them in an integrated way and learn from that. So in that context, we also formed a collaboration with a small company here in the Cambridge area called Plex, that have an AI-enabled search engine that helps us to connect our proprietary database with the outside world of, there's many databases about targets, about chemical structures, diseases. And so we can really effectively integrate all that information together and mine and find those novel connections that might not be obvious just by looking at the data in the first place. So this is also a big aspect that we had to pay attention to from day one.
No, and that goes back, at least in my mind, to what you were saying about team and being thoughtful about who you bring together, especially when you have such a phenomenal platform to build from, to draw from. That integration of tech and bio and bringing together people of both skill sets can be occasionally challenging from a hiring and a cultural perspective.
Yeah, and speaking the same language, I think that, I mean, not only, I mean, as you can probably tell, I'm originally from Germany, so I mean, you need to understand all our different accents, but the tech language and the research bio language is very different. But that's the fun. I think that's really the fun. And Lux, one of our investors, is more a tech bio company, a tech bio investor. And I mean, what really attracted them to Atavistic was this ability to create this large map of protein metabolite, this interactome, as we talked about, and to leverage that. But at the same time, also quickly be able to use the information generated for immediate use to find novel therapies that one can bring forward and make a difference for patients, because that's what we're here for, to really find new treatments for patients with a high medical need.
And you'll be able to do so as you develop this further at a faster and faster rate, which is phenomenal. And so far, Marion, Jared, you've led Atavistic from inception to over 60 million in funding in just 2 years, two candidates moving forward. Two drugs moving forward at this point, is maybe a better way of phrasing that. And so I'm sure there have been a couple of challenges along the way. Are you able or willing to chat about them today?
Yeah, I mean, there were operational challenges, because we started the company in 21 in the midst of the pandemic. I mean, and the supply chain. So I mean, hiring people, bringing them back into the office or labs, and I mean, equipping the labs, finding the lab space. I mean, it was all really challenging in 21 when we kicked off. It was greatly helped by this huge network I've built in my 25 years in Cambridge. So I had a lot of resources to tap into that helped finding space, building the labs out, overcoming supply chain shortages in creative ways. And so, yeah, no, we have overcome all of that and very quickly. The other thing is, from a more strategic scientific point of view, one of the challenges is, and I mean, that might sound funny, I mean, the platform is really working. So we have generated a lot of promising data, and where we could go in many directions, but we constantly have to prioritize what to focus on, where to invest our resources, where we think we have the most chance of long-term success. So we cannot do it all. So it feels like there's so much promise there, and every day we have to decide not to do something just to do the right thing. And so this prioritization exercise, and Jared has lived through that with us, is hard, because you just want to do it all, but because the platform is so successful and it has been validated now on many proteins, we just have to make decisions what to focus on. So it's a good challenge. It's not a bad challenge, but it's still a challenge.
Absolutely. And that's actually touches on one of the questions I had generally, which is when you have such a robust data engine, which is in so many ways what this is, it produces a large number of opportunities that can sometimes then be difficult to narrow the scope. Have you considered, or as a company, are you considering, and maybe this goes beyond the pale of the interview, but have you explored the possibility of partnering to expand what you're able to explore on your own? Is that something maybe down the line that is an opportunity you're keeping in mind, out of curiosity?
Yeah, in order to leverage the full potential of the platform, ideally we would like to find partners to work with, because we cannot do it all on our own. And there's so many exciting targets out there and so many difficult diseases to tackle. So yeah, no, we are very interested, and we're in active discussion with potential pharma partners to work together to leverage the platform at its best.
Thank you for sharing that. And again, broader question for you both. We've talked about this throughout the episode so far, but would love to hear in your own words and just ask it directly. What have been some of the greatest accomplishments so far in your mind?
Yeah, I have to say, whenever a technology is spun out of your lab, there's always a fear that it's not going to be robust in the hands of the people that catch it on the other side. And you're going to have to show up at a meeting and listen to them say how your technology doesn't work and deal with the shame of that. And, I had great confidence in Kevin Hicks and the people in the lab that built the Midas platform, but it was still scary. And, from myself, I think the accomplishment of the joy of seeing it work well in Atavistic's hands has been very gratifying. And in fact, they're doing it at a level that we could only dream of. They're doing it in a much more robust and sophisticated way than we have done it in the past. And so that's been really gratifying to see them industrialize it and perfect it and really use it with great impact.
Yeah, I mean, that's a really good point that Jared is bringing up. You have to know that when we started the company, we fully relied on the experimental data and process that Jared had developed. We did not have a seed stage, or an incubation stage. It was really all relying on the fact that we can get this platform up and running and that everything is going to hold up. So I was very pleased when that was indeed the case. 21, I was employee number 1, and we had raised our Series A with the Column Group, Lux and Nextech. And then it was about finding lab space, really getting going and making sure the platform is working. And it's a platform based on mass spec. Those are very high-tech, difficult instruments, and they were difficult to source at the time, but we got it to work, and it worked as advertised, and we were able to make great improvements on it because of the experience of our team that we have hired. So we could optimize it further, make it more scalable and more robust, as Jared mentioned. So I was very pleased that our team got that going so fast. But being a drug discoverer at heart, what I'm most excited about is that we are already in that process of advancing 2 drug discovery programs on their path towards the clinic, one in precision oncology and one in rare diseases. So I think the applicability of the platform to drug discovery, that we could show that and we could really make that reality and have that vision come true, that's very rewarding. And I'm also proud of the great interaction we have kept between our founders. I mean, Jared and Ralph and the company. We have every other week our founders call where we exchange the latest. And,
I wanted to touch on this a little bit more deeply, and if I may, Marion. Jared, press releases on Atavistic regularly mention this sort of co-founder relationship that you're describing with yourself, with Ralph, staying very closely involved with the scientific direction. As someone who's spun out a couple companies, who's taken several SAB and other collaborative industry positions, I would love to hear your thoughts about staying connected as an entrepreneurial PI. And if you have any advice to other PIs who might be doing something or thinking about doing something similar.
Yeah, I have to say, I've just had a great experience being involved with the private sector. I feel like it's dramatically enriched my scientific career. You know, these founder calls that Marion was referring to, these are fantastic scientific discussions that are inspiring to me. And I leave, you know, thinking about science in a new way. And you know, Marion has built an amazing team at Atavistic that, you know, challenges all of us, you know, intellectually each time we meet. And so from my perspective, this has been, you know, all plus, you know, it's been all positive for me. The very little costs. Of course, there's a little bit of time and none of us have as much time as we wish we had, but well worth it. And I guess I feel like, you know, as an academic, you know, I get paid to do, you know, biomedical research. And I feel like one of the outputs of that, either directly or indirectly, should be doing things that matter for patients someday. And I feel a responsibility to try to, in one way or another, translate the work that is done in my lab to, you know, to get to a patient. And from my perspective, with the kind of work that I do, that almost always goes through the private sector. And so company creation and being involved on SABs is I think, a very important part of what I consider to be my academic legacy. You know, that's part of when I look back when I'm retired, I think I will be excited about the research that my lab did and the students and postdocs that were trained in my lab. I think I will also be excited about hopefully being able to impact ventures in the private sector that hopefully will have drugs in the clinic that are benefiting patients' lives. I feel like it's just, for me, it's completely integrated with my academic pursuits. And, you know, I guess one other thing I will say about that, I've been talking with many of my academic colleagues over the last little while is, you know, cost pressures in academia, you know, our spending power in academia continues to go down and grants are not keeping pace. And Marion tells me there are also stresses on the private sector side with funding, especially these days. You know, we will be better together. You know, if we can figure out how to take the best of academia and the best of the private sector, what we are really good at, and synergize those contributions, I think we will be better together. We will be a more effective biomedical research and development enterprise. And I think that will be the way that we can maximize the impact we have on the health of humanity. And I feel really passionate that we do that okay, but I think we could do it better. And it feels like something that, you know, is maybe the grand calling of our lifetimes in a way, is to maximize our impact in that way.
And this is really well said. I think it's really this close integration of academia and industry. I mean, really, constant exchange of information and no egos involved, that you really can have this open discussion. So if we make an improvement on the technology that Jared has developed, he's happy about it. He's not like, oh, mine, it was perfect. So he's always helping us to make it better, and he's interested in that. And really having those regular founders call is very helpful, because if you just in the beginning start the company together and then you don't continue your interactions, that's a very different proposition. I think it's really constant interaction that makes it successful.
And as we think about that constant interaction and we project out, Jared, as you were saying, as you were both saying, into the future, can you tell us maybe what's coming next for Atavistic?
Yeah, I mean, we gonna continue on our path to really work on the most relevant drug targets that are out there and where we can really make a difference for diseases that have a high medical need. We want to be, in the long run, a fully integrated drug discovery company that has several approved therapies. And at the same time, you always want to maintain our cutting edge. Obviously, it's a long path to get there, so we have to be very disciplined as we move along, and really make sure that we can clearly demonstrate all the value we are creating on the way. I think given that we started really 2 years ago with an empty lab and not even a coffee machine or internet access, we have come a long way. So I think we are on a great trajectory to discover novel therapies for targets that are of high interest and of high value, and then bring them to patients and to the markets eventually.
And again, question for you both. As we think about the future of allosteric drug discovery, are there any key scientific advancements or enabling technologies that you're either hoping to bring forward or excited to see, either now or in the future?
Maybe I can just add one thing to that. I think, we talked about, Chris, about this future where we might know this matrix, right? Where we know the interactions between all proteins and all metabolites. I think if we even got close to that, to the point where we can use, I think it's very reasonable to think that we could use machine learning technologies if we had a training set that was robust and powerful. I think we could then predict a lot of what is yet to be discovered. And I think there's a future, and hopefully it will be within the lifetime of Atavistic, that we can do that we can then interrogate the evolved, small molecule binding sites on all human proteins and know which sites can bind molecules and what the biophysical and biochemical and chemical features of those molecules are. And, it's not even impossible to think that we might be able to predict off-target interactions of drugs, that hit proteins that we weren't looking to hit, and that might explain toxicity and drug-drug interactions and how drugs are metabolized and things. I think this is all part of a future trajectory that starts with where Atavistic is today. And I'm really optimistic to think about that future. And, in addition, I think you asked the question about what enabling technologies are still coming along. The platform, the Midas AMPS platform that Marion and I have talked about, there are other technologies that could be used side by side with it to discover small molecule protein interactions in a complementary way. And I think that will continue to develop. And I think there's biological understanding that we still need to build upon to leverage the discoveries that Marion and her team are making. And so I think there's a lot yet to be done, but I think the future is really bright. I feel like the trajectory is steep and the future is bright.
Yeah, and I think, again, coming back to a bit more the drug discovery angle, to optimize small molecules to make them most drug-like for, it's greatly helped by structure-based design. And just today was actually an exciting day that AlphaFold was nominated or got the Lasker Award nominated. So obviously this is a predicting of structures without having to experimentally crystallize a protein to understand the structure. I think that is the dream, and they are on their way there. I mean, in our hands so far, we still benefit from doing the actual crystallization of a protein, in particular when you want to map small molecule protein interactions. But I mean, as those technologies evolve, like AlphaFold, getting really totally predictive, and also additional computational modeling approaches that are already being leveraged by many companies as they get evolved more and more, I think that will make our job even easier and faster to develop new therapies.
A great answer, both. And normally we like to ask where you see things going and where is biotech, or in this case, allosteric drug discovery in 2050. But I think you've already started to provide an answer to that question. So thank you for preempting us. And
yeah, it's kind of like a renaissance. For me, allostery is like a renaissance of small molecule drug discovery. So it's really a great way to tackle many targets.
No, and if there's going to be a platform bringing it forward, it sounds like Atavistic is building the one. So looking forward to seeing the drug candidates and drug programs you're bringing forward. Looking forward to learning more about the success in the coming years. But for now, before we come to a close, and in that future time of success, we'd love to bring you back for another episode and talk about it further. But before we come to a close, maybe just I was thinking just a few, but hey, Atavistic is still around. It's a mainstay drug company, bringing patients forward and helping advance the science. And at that point, who knows? Maybe Jared's founded another couple companies, and Marion, you are helping run this one in collaboration. But for now, just a few rapid fire questions to round things off. Something we always like to ask. We've talked a lot about professional topics today. I'd love to ask you each something personal, which is just, what do you like to do for fun? How do you like to spend your time outside of work?
Well, you might see I have a master's jacket on, so that gives away one of my hobbies, which is to golf. That's golfing and skiing and spending time with my family are the three things that I probably most like to do outside of the lab and the office.
Maybe share it in reverse order.
That's right. Very much so. Thank you, Chris, for keeping me okay on the home front.
Yeah, just to mess with you a little bit, Jared. Marion, what about you?
Yeah, no, I like to spend time with my family. I have 2 college-age sons that just in the midst of college. So and whenever possible, we get together. We like to ski. We're a big skiing family. I haven't skied with Jared yet. For some reason, because we usually go to Europe when we go skiing. But I like to ride my bike. I ride my bike to work. And I'm a city person, so I love to go visit cities. Like at one point I lived in New York City, which was my favorite place to be. Going to modern art museums and really exploring new things.
Chris, don't you think it would be logical to have a future Atavistic meeting in Salt Lake City where we can couple the good science with the good skiing? Doesn't that make sense?
I would love to be invited, so please and thank you.
You're in.
Before then we wrap up, just any other closing thoughts, shameless plugs, anything on Atavistic, anything you'd like to share with the audience that we might have missed?
No, perhaps just to reemphasize, the AMS platform is working, and in order to realize its full potential, we're really interested to find additional partners to work with. So perhaps that's one thing to add on on my end.
Yeah, thanks, Chris. I think it's been a lot of fun to talk about this and envision the future. That's always fun to do, and it's been fun doing it with you and Marion.
Yeah, no, it was an exciting experience. So we'll see how we sound.
Oh, it's going to be great. And thank you both, Marion and Jared, for a fantastic episode today. We are very grateful for your time.
Yeah, thanks for hosting us.
Thank you very much.